All right, so here we are, Unit 11, nearly through the entire text at this point. So hopefully you've been enjoying the lectures, and we'll get together at least for one more. But let's go ahead and discuss our topic of DNA fingerprinting, again, more commonly known as either DNA profiling or genetic fingerprinting. But we'll take a look at what this entails and hopefully answer any questions you might have. Certainly reach out to me if it doesn't answer questions that you continue to have about this topic. Now, in terms of history, DNA has not been known for all that long. It was first observed back in the late 1800s by Friedrich Miescher, but he did not understand its importance at the time. But again, he was the first to record an observation of it. It took about 100 years almost to acknowledge how important DNA is for the continued existence of life and to really understand what that deoxyribonucleic acid is all about. So not a whole long time since it's even been observed, and certainly a much shorter time since it's been well understood. Okay, so now speaking of the team that did the initial understanding of DNA between 1953 and 1965, we had major contributions from James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. And in terms of the textbook on DNA, the double helix, that's credited with Watson and Crick, and they shared the Nobel Prize for their discovery. But really, a person who's missing from our text and missing from our list up here is Linus Pauling. So Pauling was a chemist, and he made the major discoveries in molecular biology in terms of protein structure, primary, secondary, tertiary, and quaternary protein structures. And of course, if you read the text makes a big deal about how proteins were believed to be the carriers of genetic information at first because they're so complex, and how could something as simple as DNA be responsible for such an important task? Well, again, Linus Pauling discovered those more complicated protein structures, but the simplicity of DNA eluded him, mainly because he didn't get to see what Watson and Crick got to see, and that was the X-ray micrographs of Rosalind Franklin's DNA X-rays. So if he had seen that, there's little doubt that our good friend Linus Pauling would have made that discovery as well. But since he was not permitted to leave the United States, he was an outspoken war critic, and he did not have his visa to attend the conference in England that Watson and Crick saw Franklin's X-ray micrographs. If our good friend Linus Pauling had attended, very few people doubt that he would have made the discovery first if he had seen those X-ray images. So Linus Pauling has the distinction of being the only person to hold a Nobel Prize in chemistry and peace, but that peace prize probably cost him a second chemistry Nobel Prize because his anti-war activities kept him from, again, attending that conference and potentially viewing those micrographs. So there we have it. Rather interesting history. That doesn't even get into the sort of soap opera Rosalind Franklin issue with Watson later claiming that they stole her work and she wasn't credited properly, but she died in Francis Crick's home, so I don't think there was too much animosity there. Again, you can't make this stuff up. If you want to learn more about it, please do look into it. It's a very interesting thing, and if you look into James Watson and the crazy things he's said since he won that Nobel Prize, you'd be amazed at some of the very unscientific comments made by this Nobel Prize winning scientist. All right, so let's fast forward a few decades and let's talk about the history of DNA with respect to forensic science. So in 1984, this is the beginning of the application of DNA technology to forensic science. So the person this time who was credited as Sir Alec Jeffreys, and he made the discovery of restriction fragment length polymorphism to individualize DNA. That's where we came up with the idea of DNA fingerprinting. Because overall, all of us as humans share an incredible amount of DNA. Over 99% of our DNA is nearly identical for all humans, but that roughly 1% that has the differences, that's where we can identify one individual as opposed to another. And that's what Jeffries was able to manipulate in order to make DNA as individualized as a fingerprint. And so we'll talk a little bit more about the particular case that he was involved with. But again, a lot of the crime case studies that we've looked at in the course all predate the mid-1980s. And so even though DNA potentially would have been a useful piece of information, it was missing from a lot of those historical cases. At least our ability to analyze it was missing. So that's something to keep in mind moving forward. All right, contrary to popular opinion, the first application of DNA in forensic science did not occur in the good old US of A. It occurred over in England, and this was 1986. DNA was used to identify Colin Pitchfork as the responsible individual for the rape and murder of 2 young girls in the English Midlands. So first, Linda Mann was raped and murdered in 1983, and then just about 3 years later, Dawn Ashworth was also raped and murdered in this small town. So the reason I say it didn't happen in the U.S. is because we tend to value our privacy a little more than the Europeans as a whole. And so in this case, the way that they were able to find Colin Pitchfork was to require DNA samples from the 1,400 residents of this small town. So again, they were forced to submit the DNA samples without any evidence against them. There was no reason to suspect them other than these are people who lived in this small town where now 2 young ladies have been raped and murdered, and they complied. Everybody in town, including Colin Pitchfork, our rapist and murderer, submitted their DNA samples. And so from those 1,400 DNA samples, the restriction fragment length polymorphism analysis was performed, and it was found that the DNA profile that matched the DNA found on the victims was that of Colin Pitchfork. So it worked. Again, you can't argue with the results, I suppose, but I doubt we'd be able to do something similar even in a small town in the U.S. Having people be forced to submit DNA samples just because they live there probably doesn't sound like anything that would happen easily over here, as easily as it did back in the mid-1980s in England. Okay, so now that we've set the scene for when forensics started to make use of DNA, let's take a step back and just respect this incredible molecule. So there's its full name, deoxyribonucleic acid. In some of the older texts, you might see dioxyribonucleic acid. That's the biologists had originally mistakenly named it as that. And if you look at some of the early texts or the biology texts from quite a while ago, you might see that. But no, chemically speaking, it's deoxyribonucleic acid because it's different from RNA ribonucleic acid because it has 1 less oxygen. So it's located in the nucleus of the cell for the so-called nuclear DNA. We also have mitochondrial DNA that's outside the nucleus. We'll talk about that. It's not as individualized, but it still is useful physical evidence. It contains our genetic information. It has the, again, the shared complements from mom and dad, and it has all the instructions for our cells to make the proteins and do the work that they need to do. It's made from 4 nucleotides, adenine, thymine, cytosine, and guanine, and it's wrapped up in a compact structure called a chromosome. These molecules of DNA can be centimeters long, so that's amazing for a molecule when we're thinking about angstrom size atoms to get something that's centimeter scale, but again, in order to keep it all compacted, we have it wrapped up into these chromosomes. Everyone but identical twins has unique DNA. Identical twins have identical DNA at birth, but they might get different damages to their DNA with time, and so that's been something that's been argued over and over again in forensics, trying to distinguish identical twins in adulthood based on small differences in their DNA. The juries don't tend to buy it. They tend to prefer fingerprint evidence for the reason that identical twins have very different fingerprints and only very slightly different DNA by the time they've reached adulthood. So that's something that I think will change. I think, as the methods get better and more accepted, we may see even among identical twins DNA evidence being able to distinguish, but for the most part, at the current state of the technology, we tend to prefer other ways to distinguish identical twins. All right, so looking at those nucleotides, so they have a phosphate group attached to a sugar group and one of 4 nitrogenous bases. So there's the nitrogenous bases that we mentioned: adenine, cytosine, guanine, and thymine are found in pairs. So adenine always pairs with thymine in DNA, and thymine is actually replaced with uracil in RNA. But if we're going to focus on DNA as we are, it's adenine with thymine and cytosine with guanine. They just happen to fit each other perfectly for the hydrogen bonding interactions that aren't really bonds, but they're strong interactions that allow the DNA to stay together when it's not being accessed, but then open up when cells need to duplicate DNA for cell division, or you need to get instructions for protein synthesis from the DNA, that sort of thing. So it's really elegant the way nature has the DNA binding with those hydrogen bonds so that it can stay zipped up when it's not being accessed and unzip fairly readily when it does need to be accessed. So these nucleotides are linked into chains to form what we know as the double helix, the classic. If you think about these spiral staircases, you'll sometimes see that's a helix structure. So DNA is a double helix because we have the 2 different sides. For an example, to see how well you're following along with this A and T and C and G stuff, if we had one strand of DNA with the sequence AAT, GCG, GCTACG, what would the other strand's sequence be? So take a moment, pause the video here if you need a moment to think about that, and start it back up when you're ready. All right, so if we think about our complement, we'd have to have TTA, CGC, CGAT, GC would be the complement, just replacing the A's and with T's, the T's with A's, the G's with C's, and the C's with G's on the other strand. So hopefully that has been useful. Okay, so let's think back to the earliest point that any of us existed, and that's when we were a zygote. So zygote is what happens when the sperm from the father and the egg from the mother meet and join and create the new being, the offspring. So that sperm carries with it 23 chromosomes, and the egg has 23 chromosomes as well, for the full complement of 46 chromosomes for a typical human being. I say typical because there are some with 47. There's certain trisomies like Down syndrome where you have an additional chromosome. And again, they're certainly still human beings. It's not a common thing, but it does happen from time to time, especially Down syndrome in particular is a fairly common example of a trisomy. So occasionally there's 47 or so chromosomes, but typically it's 46 chromosomes, 23 from mom and 23 from dad. And they have all the information that any cell of the body will need for the instructions on how to make the proteins that humans need. So again, half of your DNA was inherited from your father and the other half from your mother. And so the offspring inherit about 3 billion bases, making up around 20,000 genes. Again, it's all with the 23 pairs of chromosomes, 23 from mom, 23 from dad, for a total usually of 46. Now, we mentioned mitochondrial DNA earlier, and again, it's useful DNA. It's not as individualized as the nuclear DNA. The nuclear DNA is, aside from identical twins, as we mentioned, it's individualized. It's only a single individual in the world that would have that DNA. For mitochondrial DNA, this is going to be just down to the maternal line. So let's talk about what it is first, and then we'll talk about that restriction. So mitochondria themselves are responsible for respiration and energy production of cells, and the belief is that these were things that became engulfed in animal cells at some point in history. So there's a symbiosis. Our cells provide them what they need for energy production, and they provide us with energy, and so that's how we evolved to have mitochondria. They were engulfed by animal cells a long time ago. So if we look at mitochondrial DNA, it all comes from the mother, because the female egg keeps her mitochondria during fertilization, and the male has mitochondria. The sperm cells have mitochondria, but they're mostly in the tail, and the tail falls off as the head fertilizes the egg, as we saw in the previous slide. So that's where we end up with just the maternal line, is all of the mitochondria come from the mother's egg, none essentially from the father's sperm. So these are smaller than nuclear DNA. They are, as a result of being smaller, they tend to be fairly well conserved. They tend to be fairly robust and stable. They stay around for a long time. You can find mitochondrial DNA from mummies. Again, the main drawback is it's an exact copy of your mother's mitochondria. So it's useful in genealogy. If you happen to have a living relative along the female maternal line, then you would expect identical mitochondrial DNA. So again, it's not individualized like nuclear DNA, but it can at least get you down to a maternal line, which can be useful. The other useful feature of mitochondria is because it's always from the mother. If you're finding yourself in a situation where, there's babies all over the place, and babies are beautiful in their own ways, but sometimes you just don't know what to say about a baby. And, the thing that saved me on multiple occasions when I don't know what else to say is, well, he or she certainly has his or her mother's mitochondria. And people will sort of smile and stop talking to you, and you've made an awkward situation a bit more awkward, but at least you haven't said anything dishonest. It's entirely scientifically valid, and that's about all you had to commit to that particular baby. So if you want to use that, go for it. Again, people probably won't talk to you much more, but at least you have made your way out of that particular bind. Okay, so here's an image of those chromosomes we mentioned. So they carry the genetic information. And humans have 23 pairs of chromosomes. Again, 23 from mom and 23 from dad, for a total of 46 in a typical human cell. And they tend to all have this sort of appearance. They look like the letter X, or the Greek letter chi, really, aside from the male Y chromosome. So that's the one time you would have a smaller, more Y-shaped chromosome. And that's how you can identify males, by the presence of that Y chromosome, whereas the females, all the chromosomes will have that X-type appearance. So again, that's what we're tending to look at. One of the reasons why males tend to be more impacted by things like color blindness is because that tends to occur along the X chromosome, and so females with 2 copies of the X are more likely to have a good copy, whereas males who have just 1 copy of the X are more likely, if there is a defect, than if they only inherit 1 X chromosome, so they're more likely to be impacted by things like sex-linked traits like color vision. Now, if we look further into just genes, that's a section of that DNA along the chromosome that contains the inherited information. So, we inherit genes for good things and bad things too, unfortunately. So a lot of you have probably heard about certain oncogenes, genes that might cause cancers later in life. So if you happen to know that you have a gene that puts you at a higher risk, like Angelina Jolie, you may take some precautions. She was very proactive in had her breast removed because of her gene that had an increased likelihood of breast cancer. She didn't wait around for that. She took action in advance. So I'm not advocating that, but it's been done certainly. It's something that is a very personal decision. But based on your genes, you might find that those sorts of decisions might be right for you or maybe not so right for you. But we live in an age where at least now we can be looking for that sort of thing, and the genes that you've inherited are going to be more and more important in medicine as we get more focused molecular medicines. All right, so let's move back to forensics and let's look at potential sources of DNA in terms of physical evidence from a crime scene. So we've got saliva, or anything that's likely to have saliva residue, like the top of a bottle or the rim of a cup. Something that someone's mouth has touched directly is a likely source of DNA because of saliva. Blood, again, we talked about this a few units ago, but blood, it's really the white blood cells we're looking for the nuclear DNA, but even though they're a small component of blood, we're usually able to isolate them if we do have a blood sample, whether the blood is dried or still fresh. We have different ways of collecting those samples. Semen is another potential source of DNA for either a scene of sexual violence or any situation where you might have semen residue as a source of DNA. Sweat, skin, hair, finger, toenails, bones, and teeth with root material can sometimes be a source. Again, we say sometimes because hair, depending on if we've got the follicular tag or not, we talked about that a while ago. If it's got the follicular tag, then it should be relatively straightforward to get DNA. If it doesn't, then again, there's new methods that may prove helpful, but they're very expensive, very time consuming, and reserved only for extreme cases. So we always collect it as though it has the DNA because we don't want to damage any potential DNA, but unfortunately sometimes the DNA is not in a situation, not in a state that we can actually get useful information from it. That's why we try to collect a lot of physical evidence so that something hopefully has usable DNA from the suspect, victim, whatever the case may be. So keeping in mind those potential sources of DNA, let's talk about collecting DNA. So there's a number of different tools that can be used: smear slides, scalpels, tweezers, scissors, sterile cloth squares to collect, say, blood evidence, for instance, UV light, luminol, blood collection kits. Again, if we're talking about, say, a dried blood stain, then we could try to collect the whole item that the dried blood is on, or we could moisten a cotton swab with some saline or some tap water and blot it until we get that dried blood hydrated enough that it goes into the cotton. Again, if you use tap water there, you would need to collect a separate sample of the tap water for comparison's sake. If you're collecting DNA samples from clothing or from furniture, again, we mentioned it with dried blood stains, but with any potential source of DNA, submit the entire piece wherever possible. Again, if it's a large area like a wall or objects that are too big to send to the crime lab, then the samples are usually scraped into a plastic or a glass container. We talked about the special handling of a dried blood stain earlier. We'd like to have everything in a non-airtight paper bag because, again, molds will thrive in moist environments, so we do want to try to keep everything with any sort of biological evidence in a non-airtight paper bag to allow the evaporation of the water and prevent the growth of molds. Okay, so once we've collected the DNA evidence, now let's talk about analyzing DNA. So DNA is converted into a series of bands called DNA typing using only segments of DNA. So again, those restriction fragment length polymorphisms where we're looking for just the parts that are unique. So the method depends on the amount and the age of DNA. So if we look at, again, the RFLP that I just mentioned, polymerase chain reaction, PCR, can help us if we find very little DNA. We might use that method to magnify our sample before performing analysis. There's short tandem repeat analysis, the STRs, mitochondrial DNA analysis. Again, if it's mitochondrial DNA, it will get us at least to the maternal line, but if that's all we have, then it's better than nothing, certainly. And gel electrophoresis can help make those bands, right? Those different bands that will distinguish which particular suspect fits the DNA evidence from the scene. So let's talk a little bit about these different DNA analysis techniques. All right, so the first technique applied, restriction fragment length polymorphism, RFLP, is the one that we tend not to use so much anymore. So the reasons are it requires a large amount of DNA. So back when they were sampling the 1,400 residents of that town in England, they had access to large amounts of DNA, so it was no problem. But unfortunately, in forensic science, usually now, especially since that early days where DNA became obviously a type of physical evidence that could match a suspect to a scene, like it did with Colin Pitchfork, most suspects don't leave around a lot of their DNA. They know better nowadays. So this method's not ideal because usually there's only very small amounts of DNA found at crime scenes, and again, this just doesn't lend well to RFLP analysis. It works when there are the required amounts of DNA present, but again, it's not typical in forensics anymore. Okay, so polymerase chain reaction I mentioned briefly before. This is a way that we can magnify a DNA sample from a scene if we have, again, that usual situation where only very little bits of DNA are present. So this tries to mimic what happens in the body, right? Our DNA replicates itself all the time so that each new cell will have its own complete copy of DNA. So we do this a little faster here by heating the DNA up to around 90 degrees Celsius. That makes it unwind, and then you get the two new duplicate strands are made from the polymerase. Taq polymerase is the one that's typically used. Thermophilus aquaticus, a heat-loving type of polymerase, right? Human polymerase doesn't work well around 90 Celsius. It works well around 37 Celsius. So we have to reach out to other species to get something that works well at those high temperatures. And again, if you go too high, you can have problems here where things won't work. You certainly don't want to boil the sample with the DNA in it. So keep doing this sort of process for around 2 hours, making sure that you have enough of the nucleotides and everything to make new copies of the strands, because you're going to make a lot of copies. So in 2 hours time in a DNA amplifier, you can take that small amount of DNA and make millions of copies. So that's pretty amazing that we can do this. And again, it solves that problem that I just mentioned, where we don't tend to get a lot of DNA at the crime scene, although there are potential problems, and you have to be very careful about contamination here, because certainly the defense is going to argue that there's contamination issues if there's any hint of impropriety in how this technique was analyzed. We want to make sure that without doubt it's just magnifying and not altering the DNA from the crime scene. So even with the ability to magnify using PCR, we still tend to use a different method than that RFLP, which again, we said the problem is it requires a lot of DNA, but then we just said that PCR can make more DNA copies. Well, the reason we tend to do this short tandem repeat analysis is that it can be done with a very small amount of DNA. And again, we can magnify our sample, but we also have to be careful, as I mentioned, of contamination issues or mutations happening in that DNA that's magnified. So we don't want to overdo it with the PCR method. We do enough to get an analysis of DNA that allows for individualization, but again, it seems like this short tandem repeat analysis does a better job with less DNA than the RFLP method from early on. And going back to mitochondrial DNA analysis, we can use this for very old or degraded DNA. And so we can talk about hundreds of years old or even thousands of years old DNA now. It's not going to put anybody behind bars, so to speak, but at least it can help us get to the bottom of historical cases where we're not sure, whose body is in which grave and that sort of thing. So for instance, this technique was used to identify Tsar Nicholas II of the Romanov family decades after his assassination, again by using a living relative along the female maternal line and testing the mitochondrial DNA of that person as well as the person believed to be Tsar Nicholas' body. The same sort of technique has been used to identify Jesse James, who was an American outlaw from the 1880s, using mitochondrial DNA from his teeth to determine if it was his body in his grave. That case has a lot of twists and turns, so if you want to have a really interesting read, you should definitely look into that Jesse James situation. They exhumed his body, they had a living relative along the maternal line for a mitochondrial DNA sample, and they said that they had a match, that it was Jesse James in his grave. And then the sheriff said, well, I never actually took the DNA sample from the body. It was, I think it was from himself. The sheriff himself gave it because he didn't like disturbing the Jesse James body. And so then it called into question the researchers, and I guess it was all finally resolved to say that it was indeed Jesse James' body, but there's enough doubt in that situation that if you'd like to read more, I would definitely recommend you take a look at the Jesse James case for some more interesting study of DNA analysis. Okay, so for that analysis of DNA, oftentimes electrophoresis is employed. So this is a situation where you make a gel, and that gel has certain wells into which you inject the samples, and the fragments of DNA from those wells move through the gel by electrical potential. They'll all be coated in a soap, usually sodium dodecyl sulfate or some soap that gives them all a negative charge, and then they migrate from the negative end to the positive end of the electrode potential that's been set up there across the gel. And so it should be the larger fragments that move less, and the smaller fragments can move more easily through the gel, and that's how we separate based on size, mainly because we've given them all the same negative charge. So the bands can be stained so that they fluoresce, and then compared side by side to see if there's any bands that seem to be identical. And so we see in our image here, we've got the first and last band seem to match up pretty well, right? And the second and the second from last seem to match up pretty well. And so again, the small differences we have in our DNA will allow for us to distinguish between humans, even though we share 99% of our genes. These small differences will allow individualized analyses. And now to wrap up our topic of DNA fingerprinting, let's talk about CODIS. So CODIS is an acronym for the Combined DNA Indexing System, and so that's the FBI's software and database and all of that good stuff that is used to support the national DNA indexing system and the various state and local level databases. So the NDIS that I just mentioned, National DNA Indexing System, is part of the CODIS, and it has two groups in there. There's the Convicted Offender Database, so depending on the level of crime, a convicted offender may be required to submit a DNA sample and be a part of this database. There's also the forensic files, which is DNA from crime scenes that hasn't been matched yet. As I mentioned earlier, a lot of the historical cases we've studied have occurred before the application of DNA technology to forensic science. So some of those old cases have been looked back at through things like the Innocence Project, and we've been able to exonerate innocent people. There's the Central Park 5 case that is a pretty big deal in terms of 5 young men who through forced confessions were convicted based on spotty evidence, mainly hair evidence that sort of overstated what could be done. And then it was later found that the DNA from the jogger there who had been raped and attacked, it was not a match for any of the men, and so they've been exonerated. One of the co-founders of the Innocence Project was one of the defense team for O.J. Simpson back when, again, that case was tried. It was an early application of DNA to forensics still, and so he was an expert in that area, and then later went on to try to exonerate innocent people based on DNA technology that had not been around at the time that they had been tried. So again, hopefully you've enjoyed this topic. It's a really interesting thing, not only for the forensic applications, but for human health and genealogy purposes, and DNA is just an interesting topic. So hopefully you found this to be a useful lecture. If there's still questions that you have, please do reach out to me, send me an email, and let me know, and I'll be happy to offer my advice and opinions, and hopefully good scientific evidence if it's a question I can answer. If it's not, then I'll certainly be upfront about that, and maybe we can learn together. So hopefully you've enjoyed Unit 11 here, and look forward to seeing you one more time for Unit 12. Take care, everybody.